introduce you, Eliza, before we...
Is that all right? All right. Our next speaker is Dr. Eliza Hawkes. Dr. Hawkes is a medical oncologist and the clinical lead in lymphoma at the Austin and Eastern Health. So Eliza's going to talk to us today about the latest information on clinical trials in lymphoma. Thank you, Eliza. So I didn't really want to overlap.
No, it's not working.
Too much with Joel and his discussion of the new therapy. So I thought I'd just talk a little bit more about what clinical trials actually are and why we do them and what you should know about them. I run the lymphoma clinical trials program here and at Box Hill Hospital. And we have a mixture of studies that we get in from pharma that we run locally for them, but we have a very big program of research that we design, develop and run ourselves, which takes an enormous amount of resources and funding and everything else, and we kind of get those from various sources. The clinical trials program is a small part of our service. We have dedicated physicians who specialise in lymphoma, who see the patients in our clinics. We have obviously Tanya, who basically herds us and tells us where we should be and what we should be doing, and also you. And then we have our trial staff as well. So clinical trials, what they really are is a whole bunch of studies that show new ways to either detect, monitor or treat any medical illness. So it can be a new test, it can be a new type of treatment, it can be testing more treatment added to the standard treatment, or it can be trying to do less treatment and, because we kind of have built up over time with more and more, but then trying to back off and get the balance right. It can be a different way to monitor patients, either with new blood tests or scans or less scans. So it's a whole bunch of different things all under the one umbrella. These are absolutely essential to improving the outcomes for our patients. Almost every single treatment that we use in clinical practice to treat cancer has been because of results from clinical trials. And cancer is one of the biggest and probably one of the earliest areas where we used what we call evidence-based medicine. And not only do we use it in our clinical practice, but the PBS, to choose which drugs to fund, use the results from clinical trials. And to develop new research, we use the other results of clinical trials that we've heard about before. So there is a very large global community of which the Austin is part. And we share our results, we share them freely, there's no secrets. We help each other design new clinical trials. It's not a big kind of evil conglomerate sitting behind going, how are we going to rip off society? And we only use proven treatments from clinical trials in our clinical practice. And I think that's really important because we are bound by those regulations, and the trial nurses will tell you they are almost weighed down by regulatory requirements in clinical trials. But if I quit my job in Western medicine and went down the road and turned on my tap and said, the water running out of here cures cancer, I can say that wherever I like, as long as I'm not saying it as a medical practitioner. And there are a lot of alternative medicines which are unproven but claim quite a lot that are not adherent to these strict regulations. And Tim Minchin once said alternative therapies that are, well, alternative medicine that is proven is called medicine. And I think, this is where it all comes from. So I think it's really important to know that the overall aim and why there's all this regulation and why we do it is to prove that a new test or treatment or way of doing something is, number one, safe, and number two, it works. And sometimes we test those two things at once, and sometimes we have to test safety first, and then efficacy. Or in sometimes we are testing if it's more safe or more effective than our current treatment. So we really need a balance of people in clinical trials, so we need different ages and ethnicities and genders so that we know that it applies to everybody, because there's no point in testing something just in women and turning around to a man and saying, pretty sure it's going to work in you, but we haven't really tested it. But it's also important to make sure that the group within a clinical trial is similar enough that you can actually say that the outcome is because of your intervention rather than because of a difference. So if you've got a bunch of 90-year-olds and then two 50-year-olds, and the 50-year-olds do a lot better, and you say, look at my treatment, that is amazing, whereas actually it's probably just that you've got quite a lot of people of one group and not another. Not to say that nonagenarians are not going to do exceptionally well, but it's just a bit different. So we have to make sure that the benefits, or in fact the side effects, are not due to differences in that person. So we can't make everybody the same, but we have to make it similar. So this is what we all have to do in order to go from the design kind of ideas perspective right the way through to registration, which is kind of government support, and that circle is the bit that we ask you to participate in. So before and after we're actually speaking to patients about clinical trials, there is a ton of other stuff. So we need an approved protocol, which is basically a set of strict instructions that we have to adhere to, and then the sites, individual sites are selected. So either we get approached by companies and say, look, would you like to run this study here? We've got an excellent reputation at the Austin for clinical trials, so we get approached quite a lot. And then we have to get local approval. So the team at the Austin has to actually approve it. And so our ethics committee looks at that to make sure that it's relevant to our patients, because some of these trials are designed overseas and they have slightly different rules. And then we open the study and we recruit, and I'll talk a bit more about that, and participate. And as that's going along, the nurses and the doctors are entering data, and we're talking to the team that runs the trial over, globally if it's an international study, making sure that our experience is the same as everybody else's, and Joe Blow over here is not getting terrible toxicity at his institution and we're not seeing any, so we're telling everyone that we're doing great. So we feed information back, they ask us for more information, and we feed more and more back. And then the trial results are all analysed, and we talk about that, and it's presented, and we kind of go to these meetings, and I'll be in Atlanta for 5 days at the end of next week listening to a lot of these, so that I can then bring that back to the team here, and we can decide not only what research we want to do or how relevant our research is, but what we think is best for our patients locally and where things are headed with those drugs that Joel mentioned before, and other new ones. And then what happens is, usually if it's a really good result and it's going to change practice, then that drug is registered with the government authorities for use.
Could I just ask a question there? What percentage of clinical trials are actually abandoned because the drug's found either not to be safe or effective?
Quite a high proportion. So I'll talk a bit about the phases of clinical trials, and I'll talk about which actually get through. So this was found possibly to be the first ever clinical trial. It was a doctor called James Lind who had these patients with scurvy in 1747, and he split them into groups, and he gave some cider, some kind of elixir of vitriol, I'm not really sure what that is, I think it was stomach contents, vinegar, seawater. Luckily some people got oranges and lemons and a few got barley water, and he figured it out, and now no one gets scurvy because we all eat our citrus fruits. So clinical trials and research in general goes in these phases, and lab studies are all of our basic scientists, and we've got the Olivia Newton-John Cancer Research Institute upstairs with our scientists all working away, and this is usually unfortunately we do have to do some research in animals because it's not just makeup, it's other things, so we have to do those tests. And we also do what we call cell lines, which is where we actually take some cells of the disease that we're looking at. So we take some lymphoma cells from tissue or from people who have agreed to give them to us, and they grow those in either a medium or, you know, kind of fancy petri dish, and we do lots of tests on those. We squirt some various drugs and the cells either die or they don't. So that's what we call preclinical, and that takes quite a long time to actually prove, and we use new combinations of drugs in those situations, and you know, we do all of that in the lab, and some of us work either with the lab scientists or as lab scientists as well. And then if we find a really promising scenario in a certain type of disease, we do what's called a phase 1 study. And some of these are what we call first in human, and some are dose escalation studies. So we're trying, now that we've found this compound, to get the dose right in people, and also a little bit of efficacy. So we're testing to see whether these drugs work all the time in the person who's on that trial, but we're also very closely monitoring them for safety. So they often have a lot of blood tests so that we can make sure, and because we don't really know a lot of the side effects of these drugs that we find in the lab, so we're learning about those. So that's a phase 1 study. About 10% of the drugs that go into phase 1 actually get to the very end. So it's a lot of work, and it's a lot of time and a lot of failure. We usually do phase 1 studies in people who don't have any other options, and that's to try and use the drug which biologically looks like it probably works in these patients, but they don't have any of the standard options left. And you know, we're testing the safety, but we're hoping for a response. We then have kind of phase 1/2. That's a little bit of overlap, because then we learn a little bit about the dose that we want to take forward of the drug and how we want to give it and what the side effects are in a smaller number of patients. And then we do that study in a bigger number. And often in those patients, we're looking more at whether it's effective and kind of collecting that information. And phase 2 studies, we don't usually give to a broad range of people. We usually narrow it down to the diseases that we've seen, you know, kind of promising efficacy in the first studies. And then phase 3 is usually hundreds or thousands of patients, and we are directly comparing that often with the standard treatment to see if it is better or at least the same. And so those ones take a few more years, mainly because we're collecting data on more patients, but also we need longer follow-up to see whether that benefit is there or not. And in something like an aggressive Hodgkin lymphoma, which generally has very good outcomes, and you know, over 95% of people are alive at 5 years, you've got to wait at least 5 years to see if there's more than a 5% difference in the newer drugs versus the older drugs, and you also need a lot of people. So even within these phase 1, 2, 3, I just thought I'd kind of talk a little bit about some of the trial designs that we use. And you may have heard of some of these phrases, but you know, a single arm study is usually phase 1 or 2. It's where everybody gets the same treatment. So there's no flipping of the coin to see whether you get the normal treatment or the drug. Adaptive trials are where we give everybody the same, and then something happens, like you have a scan or a blood test, and then we decide if the result is one thing, we'll go one way, and if the result is another, we'll go another way. Randomized control trials are what we just talked about, the phase 3, and the treatment is randomly determined by a computer. It's a bit fancier than flipping a coin, which is actually what they used to do, but it takes into account all of the different characteristics of that person, so that you don't have accidentally all the men in one group and all the females in another. So it what we call stratifies for key bits of information. Open label means we all know what you're getting, even if you're in one arm or the other. And blinded means that nobody knows. So you're right. And placebo-controlled means that there is a pretend drug and the real drug. So some people are getting the pretend drug, some people are getting the real drug. And if you're asked to participate in a clinical trial, this should all be, kind of told to you in there. We do have some newer types of trial designs now, and I've very complicated diagrams. I don't seem to have a pointer, but biomarker analyses are where we test for a specific abnormality in the cancer or in the lymphoma, and if you have it, then you will go on the trial, and if you don't, then you won't go on the trial, or vice versa. If you don't have the abnormality, then you will. So often we have now what we call a two-tiered consent process, where we say, look, first of all, don't worry about all the nitty-gritty details about the trial. Sign this and we'll get the biopsy tested, and then we'll talk to you depending on whether your result is positive or not. And we're doing a lot more of those biomarker studies because we're learning a lot more about the different types of lymphoma and how they behave. And because of that, we're then targeting some of these abnormalities in the cells. So often what will happen, is there a clinical trial for you? You really have to be eligible for a clinical trial. They have very strict inclusion and exclusion criteria. And even if we really think you're eligible, or you would benefit from a clinical trial, if you don't meet one of those eligibility criteria, then there's absolutely no negotiation. And a lot of that is not in your control. It's your kidneys aren't doing what they should be to meet the trial criteria, or your liver function's a little bit damaged from previous chemo or the lymphoma, or your heart's not working. It's mainly about keeping the people on the trial really safe, because if those organs aren't absolutely working to their maximum capacity, then we don't know whether the trial drugs are going to damage those even further. And as I said, if you fit the eligibility, usually one of us will talk to you about the clinical trial, and we have a meeting. We don't kind of just let the junior doctors hand out information about clinical trials to people willy-nilly. We will talk about whether somebody, in our lymphoma meeting, whether someone fits those characteristics for a trial that we're running, or even a trial at another institution, because we do work with everybody around town. And then we would usually come and talk to you and introduce you to our dedicated trial nurses, who are specialist lymphoma nurses who would also do the research side of it. And the eligibility is a long list of criteria that's written in medical language for us, and it usually includes a combination of these things, and I've talked a little bit about it, but the type of cancer, the stage of the cancer, the types of previous treatment that person has had, and also the test results, and your other medical conditions and what treatment you need for those. And if you are interested after a discussion, then you usually get a written document which is between 20 and 40 pages depending on the study, which will tell you about the study. These consents are getting longer and longer, and some of us think too long and too detailed, because all of that information may not be relevant to you, but that's the way the regulatory system has gone. And then you'll come back, we'll arrange for a time for you to come back to sign that. We're not allowed to do any of the research procedures on you unless you've signed that consent. We can do standard things, so some people need a PET scan or need some blood tests anyway before they have the standard treatment, so some of those we use as information for the research, but we can't go and do some wacky research test on your tissue or on you unless you've agreed to do it. So what is involved? This is really for you. We do the trial pre-screening. So as I said, in our lymphoma meeting, we usually identify people in that when we're discussing our cases. We discuss the study between the patient and ourselves, and then we give the written information, and then you come back and you sign consent. You can't sign consent on the day, you have to go away and think about it. And then after that consent form has been signed, that person then undergoes what we call a formal screening test. So we submit the blood tests and the scans and the diagnosis and the biopsy result and all the rest. And then you're either eligible or you're not. And as I said, there's no negotiation really. And if you're not eligible, then your doctor will talk to you about what your your standard options are. If you're confirmed, then you'll get a date to start your study treatment. and you'll finish that study treatment at some point, and then you have follow-up, and that's all part of the trial, but most of that is with your usual doctor or one of the team in your practice. And there are specific ways to follow up, but we're looking for often the same things as we're looking for in people who are not on a study. So has the lymphoma come back? Is that person well? Have you still got side effects from the treatment you've had? All those kind of things. So a lot of the standard tests and those from the trial overlap. And the main difference is we send the information to the people running the study, but it's all anonymised. We don't send your home number and your date of birth and your Medicare details and your full family tree. So, you know, it's all anonymised. You get a study number, and that's